So it looks like people are still coming in, so we'll take a couple of minutes and wait and then get started. Okay, so Martha, are you ready?
Yes, I am ready.
You're unmuted. Awesome. So, okay. Hi, everyone. Welcome to the very last NextGen seminar of the year. We're very glad that you're here with us today. Before I start, I want to thank our co-host, Momoko Watanabe, who has turned her video off, but she's here hiding. This is Momoko's, there she is. Hey. Wave to the camera. This is Momoko's first year as a faculty here at UC Irvine. And Momoko, I really think, I'm really grateful for you for helping host this. I think I got to you basically your first day on the job, right? And I came to Momoko and I was like, hey, I think I need some help. Can you help me host this thing? And it's really just been awesome to have you here. And so I really appreciate the work you've done with this. We've started the NextGen seminars last year as a response to the COVID shutdown. And last year, everyone was sent home, at least here in California, but I think everybody here in the room was sent home, and it was just absolutely insane. There's no toilet paper. Everybody started wearing masks. The movie Contagion, I remember sitting down and watching it, and it was like real life, and then it was like movie, and like what's real and what's not real. And like everyone else here, we had to shut down all of our projects. And, we started having lab meetings on Slack and Zoom. We didn't even know what Zoom was, right? If you think about it, like what is this? How does it work? I still sometimes don't know how it works. And we were all glued to the TV watching COVID spread around the world. At least I was, and many of the people in my lab. And so I spent a lot of time thinking about ways that we could keep the lab engaged with each other and with their research. And we went back and forth on my Slack channel about a couple of different ideas. The first one was that we could use the downtime to catch up on old seminars. And YouTube is really like a treasure trove for these types of things. I'm sure many of you have seen a number of like really excellent seminars. And so I briefly posted a collection of the seminars on our lab website, but it really wasn't what we were looking for because YouTube basically already exists. You don't need to, recreate what already exists. And so we decided to try hosting seminars. And sort of the goal was to specifically support trainees in the epilepsy field with the literal idea that COVID sucks and maybe we can just all have lab meeting together. And, there weren't many opportunities, at least at the time out there, for epilepsy trainees to share their work. And so I literally just made a Google form, slapped Jan's iDisco brain on the top of it, and we posted it to Twitter. And I think we've had a pretty successful year. I think all the talks have been really awesome. And so thank you, Momoko, for everything you've done to help with this. Thank you to everybody who's been attending these talks and especially to the speakers. I think collectively we've kind of made it through the year and have had sort of a great time to share our research with each other, sort of in a lab meeting style. And so that's been really encouraging and a great thing, I think, for the field. And so with that, I need to introduce our final speaker of the series. Today we have Martha Streng here with us. Martha is currently a postdoc at the University of Minnesota, where she's working in the lab of Esther Crick-Magnuson. Many of us know Esther from her time here at UC Irvine. And so we're making multiple circles and loops back here. And so before her time in Esther's lab, Martha got a PhD in neuroscience, also at the University of Minnesota. And she's really an expert in cerebellum, particularly in cerebellum electrophysiology and function. And she's published a number of high quality papers in these areas, both as a graduate student and as a postdoc. And so we're very happy to have you here, Martha. I'm very excited to see your research, and go ahead and take it away.
Great. Well, thanks, Bobby, for the introduction, and thank you so much for the invitation to share some of my research at this really awesome seminar series. It's been such a great opportunity for different trainees and folks to have an opportunity to present their research, and so I feel really fortunate to be able to be included in such a talented group of researchers. So I'm going to talk today a little bit about the research that I've been doing in Esther Crick-Magnuson's lab. And a major focus of the Crick-Magnuson lab is the relationship between the cerebellum and the hippocampus. And in particular, my project focuses on cerebellar modulation and how it can be used to attenuate hippocampal seizures in a mouse model of temporal lobe epilepsy. And while the cerebellum is not typically associated with epilepsy or seizures, it's predominantly considered a motor control structure. It was actually a target of early interest for therapeutic intervention to treat epilepsy. This came from a lot of work in different animal models of seizures, where different folks showed that electrical stimulation of both the cerebellar cortex and the deep cerebellar nuclei could successfully attenuate seizures in a couple of different foci. And this early work actually led to the creation of some human clinical trials. But unfortunately, the results of those clinical trials were somewhat inconclusive. So sometimes electrical stimulation could attenuate seizures, sometimes it would have no effect, and sometimes it would actually make the seizures themselves worse. So as a result of those clinical trials, the cerebellum kind of fell out of favor as a target for therapeutic intervention. But since those studies, we've made a lot of improvements in terms of the specificity of those types of interventions. And one intervention type, where what we work with in the lab, is more of closed loop intervention. So most early electrical stimulation studies did open loop, kind of constant electrical stimulation. But what we work with in the Crick-Magnuson lab is this closed loop approach. So we work with this mouse unilateral intrahippocampal kynic acid model of temporal lobe epilepsy, where you have seizures that are originating in the hippocampus. So we have an electrode that we implant in the hippocampus itself, and we record that signal continuously into a custom seizure detection algorithm that basically we train it to identify the presence of seizure events based on different characteristics like the frequency and amplitude of ictal spiking. And so then that seizure detection algorithm can detect seizure events in real time. So the stimulation is delivered in an as needed basis rather than an open loop fashion. So the seizure detection triggers the delivery of this stimulus, and the stimulus that we're using in particular also gives us an added layer of specificity. So we are working with optogenetics to deliver our neuromodulation. So optogenetics allows for specificity in terms of both the cell types that are activated and actually the direction of modulation. So we can increase neuronal firing using channel rhodopsin, the excitatory opsin, or we can decrease neuronal firing using halorhodopsin, the inhibitory opsin. So Esther worked for her postdoc, she worked on helping to develop this closed loop on-demand optogenetics setup, and she applied this approach to the cerebellar cortex, in particular, the primary output neurons of the cerebellar cortex, which are referred to as Purkinje cells. So in this work, she used the same kynic acid injection into the hippocampus. After this injection, chronic spontaneous electrographic seizures emerge over a period of weeks. These mice are subsequently implanted with optic fibers in the cerebellum and twisted wire electrodes in the hippocampus for recording the seizure activities. And these mice are bred to selectively express those opsins in cerebellar Purkinje cells. So we have mice that are expressing either channel rhodopsin in Purkinje cells or halorhodopsin in Purkinje cells, so that when we shine the light on the cerebellar cortex, we can activate or inhibit those neurons. So once the animals recover from implantation, they are set up in this closed loop seizure detection algorithm that I just described. So we record the hippocampal LFP on a 24-hour-a-day basis, and we detect those events in real time, and we randomly select about 50% of those events to receive that pulsed light delivery to modulate cerebellar Purkinje cells. And what Esther found is that that on-demand activation of Purkinje cells was able to robustly attenuate these hippocampal seizures. So these are two example seizure events that I'm showing here. The top row is one of those seizure events that was elected to not receive light, and the bottom row is one of the seizure events that was elected to receive light. And the gray bar is the time of detection. So that's the time at which that custom seizure detection algorithm determined, yes, indeed, this is a seizure event. So the blue light, which is delivered here, really quickly attenuates that ongoing hippocampal seizure. this is what the effect looks like at an individual animal level. So this is looking at post-detection seizure durations. And the hash bars are those post-detection seizure durations for the events receiving no light, and the blue bars are those receiving light. So you can see that well over the majority of the seizure events that were receiving light are stopping within 5 seconds. So this was looking at Purkinje cell excitation, and Esther also looked at Purkinje cell inhibition using halorhodopsin. And what was particularly interesting is that inhibition of Purkinje cells in this closed loop on-demand fashion was also really successful at terminating hippocampal seizures. So in this particular case, as long as you get the timing of the intervention appropriate, you can either excite or inhibit Purkinje cells and still successfully stop hippocampal seizures. So these results really do suggest that yes, indeed, the cerebellum can be a potential candidate for therapeutic intervention in temporal lobe epilepsy. So getting to this question of excitation and inhibition of Purkinje cells, this was something that was really puzzling to me as a cerebellar physiologist of how can you, bidirectionally modulate Purkinje cells and get the same kind of net effect on seizures. So Purkinje cells, this is a kind of simplified schematic of the cytoarchitecture of the cerebellar cortex. Purkinje cells are shown here in black, and these neurons are actually GABAergic. So they project to and inhibit neurons in the deep cerebellar nuclei. So one possibility is that increase in Purkinje cell firing results in a decrease in neuronal activity in the deep cerebellar nuclei. And so, the idea would be if you could go in and directly inhibit those neurons, maybe you could get the same effect on the duration of hippocampal seizures. But another particular aspect of these deep cerebellar nuclear neurons is they exhibit rebound firing. So whenever you get a pause in Purkinje cell firing, you get an increase in the firing of deep cerebellar nuclear neurons almost immediately after that pause, or during that pause rather. So that's another option, is that maybe that Purkinje cell inhibition produces a pause in Purkinje cell firing, and then you get a net increase in the firing of deep cerebellar nuclei. So we first wanted to move downstream of Purkinje cells in the deep cerebellar nuclei to try to see if we could figure out what might be going on in terms of the net effect of the Purkinje cell modulation on the activity of the nuclei. So the deep cerebellar nuclei, there are 4 different nuclei, and I focused on the fastigial, which is the midline deep cerebellar nuclei. These receive inputs from midline Purkinje cells along the vermis, and the rationale for selecting that target is the seizure intervention when delivered to the midline cerebellar cortex was particularly effective at stopping seizures. So that is what we decided to work with in the fastigial. So first, I decided to test out this idea that that increase in Purkinje cell firing that causes decrease in neuronal activity in the nuclei might be underlying seizure cessation. So I used a similar approach in terms of the on-demand optogenics, but instead of working at the level of the cerebellar cortex, I moved downstream and put my optic fiber in the fastigial nucleus. And the mice that we are working with, we are selectively expressing the inhibitory opsin halorhodopsin in VGlut2 positive neurons. So these are glutamatergic neurons, and these are the primary glutamatergic projection neurons in the fastigial nucleus. So we would be inhibiting this population. So same approach in terms of the chinic acid injection, implantation, and the delivery of those on-demand interventions. And really surprisingly, what we found is that the inhibition of fastigial neurons really had no effect on the duration of hippocampal seizures. So again, these are my 2 example seizure events. So there's really no difference in the duration in the events that are receiving light versus not. And we tried several different stimulation parameters. So the first one that we tried, this is long pulses of light, so 1000 milliseconds on, 50 milliseconds off. These are light interventions that were effective when targeting Purkinje cells, and we see no effect on the duration of those hippocampal seizures. So this is failing to produce any significant effect on seizures. We also tried shorter pulses, so 50 milliseconds on, 100 milliseconds off for 3 seconds, and even higher frequency pulses. So this would be 50 milliseconds on, 50 milliseconds off. And we didn't see any effect on the duration of hippocampal seizures. So really, we tried to inhibit these neurons in several different ways, lots of different stimulation parameters. We tried both the ipsilateral and contralateral fastigial nucleus, and we never saw any significant effect on the duration of hippocampal seizures, either at the individual animal level or across the population. So it appears as though that inhibition, that direct inhibition of fastigial neurons is not underlying the effect that we see on hippocampal seizure duration. So we next tried the opposite, obviously, excitation. So again, similar approach where we're inducing now the expression of the excitatory opsin, channel rhodopsin, in those glutamatergic neurons, and then we're implanting our optic fiber over for the vestigial. And what we see is that excitation of the vestigial nucleus really robustly attenuates hippocampal seizures. So that seizure event is terminating right away when we deliver our pulsed light. It's effective for long pulses of light. So again, looking at this histogram, we have over 90% of seizure events are stopping within 5 seconds when we use that excitation of vestigial neurons. It's also effective at several different stimulation parameters. So those are those same 50 milliseconds on, 100 milliseconds off pulses, and those higher frequency pulses. This was effective when we targeted the contralateral vestigial nucleus, the ipsilateral vestigial nucleus. Every stimulation parameter that we tried, we found to be significantly effective at controlling these seizures. And what's very interesting and I think important from a clinical or translational potential perspective is that our interventions could be very, very brief and still produce the effect on those hippocampal seizures. So this is looking at one single 50 millisecond pulse of light delivered to the vestigial nucleus, and you can see that it still significantly attenuates hippocampal seizures, such that well over the majority of those seizure events are still stopping within 5 seconds. So even these really brief interventions applied to the vestigial nucleus can have a really powerful effect at influencing hippocampal activity in the sense of stopping those hippocampal seizures. And this is looking at the population. We see a significant effect of just that single light pulse delivered to the vestigial. So this was really exciting in terms of highlighting the importance of the direction of modulation of your intervention. So Esther's work with Purkinje cells shows that the timing of intervention can be really important. And then this work is showing that the direction of modulation is also critical. So whatever our interventions are, we need to produce an excitation of the vestigial nucleus to get seizure control. So in addition, we also looked at improving the specificity of our interventions. So I mentioned that those primary output neurons of the vestigial nucleus are predominantly glutamatergic, but there are also several different types of neurons in the vestigial, including local GABAergic interneurons and local projection neurons, and then glycinergic projection neurons, as well as interneurons. So we have kind of a soup of some different cell types going on in the vestigial. So we wanted to ask whether targeting the vestigial nucleus more broadly versus targeting it specifically to those glutamatergic neurons might have a different effect in terms of the controlling those seizures. So what we did is we used a viral approach to achieve our expression of opsins. So for targeting vestigial neurons more broadly, we took black 6 mice and we injected them with a virus under the CAG promoter, which is going to be a pancellular promoter. It should get opsin expression pretty much everywhere in the vestigial, and we did get good expression throughout. So this would be exciting vestigial neurons more broadly. And then we took a more targeted approach by taking VGLUT2 Cre mice, which express Cre in glutamatergic neurons, and we injected them with a Cre-dependent virus to get our opsin expression that is restricted to those glutamatergic neurons. And what we found is when we use these viral techniques to excite these neurons in an on-demand fashion, we found that, yes, both approaches were effective. So in both cases, we were able to significantly attenuate hippocampal seizures. So both broad activation of the vestigial and of those glutamatergic neurons is sufficient for seizure attenuation. However, the more targeted and specific approach of activating those glutamatergic neurons actually does a better job at reducing the duration of hippocampal seizures. So we get a significantly higher reduction in our hippocampal seizures when we're activating glutamatergic neurons of the vestigial versus activating the vestigial nucleus as a whole. So this highlights that the specificity of your interventions is also highly important in terms of the actual outcome itself. So what have I shown you so far? I have shown you some of Esther's work indicating that modulation of cerebellar Purkinje cells can robustly inhibit hippocampal seizures. I've talked to you about some of my work in the lab showing that the direction of modulation and the cell types targeted are critical for those interventions when we're looking at the fastigial nucleus. So again, inhibition of the fastigial nucleus in this closed loop fashion fails to have any effect on hippocampal seizures, whereas excitation of those fastigial neurons can really, really robustly attenuate hippocampal seizures. And we think that this difference, this really stark difference, may explain some of the previous mixed effects seen with electrical stimulation. So, one kind of working hypothesis is perhaps those early electrical interventions that, whatever their net effect on cerebellar neuronal activity was, that they produced an increase in fastigial firing. Those were the ones that produced a strong effect on stopping seizures, whereas all the other interventions that produced either inhibition or no net change in fastigial firing, those would be the less effective interventions. So we think this has really highlighted the cerebellum as a promising target for therapeutic intervention in temporal lobe epilepsy. It also shows that increasing the specificity of the cell types targeted improves the efficacy of the interventions. So in the case of targeting the glutamatergic neurons versus broadly targeting the fastigial, we're actually targeting a smaller population of neurons. So we're actually producing you could think of it as we're producing a smaller perturbation of ongoing cerebellar activity, and we're getting a better therapeutic benefit. So that's why these tools like on-demand optogenetics are really, really important for conducting this type of research into exploring these new therapeutic interventions. And the value of those interventions is it can really say that we can get this really fine-tuned control and get an even more positive outcome. So the big question is, how is the fastigial nucleus able to influence hippocampal activity in such a profound way? And there are no known direct monosynaptic connections from the fastigial nucleus to the hippocampus. And I would say in my viral data, we have never seen any fastigial fibers in the hippocampus. So that means there must be at least one intermediate brain region that's modulating or that's mediating this effect. And so looking downstream from the fastigial nucleus, if we take those virally injected mice and, you can see the cell bodies in the fastigial, you can also see their axonal projections and see where those fibers are terminating. And it turns out the fastigial is projecting to a whole bunch of downstream regions. Three that jump out right away to us as we were doing this work are the superior colliculus, which is here in C, the reticular formation, this is the portion in the medulla that I'm showing in D, and then the centrolateral nucleus of the thalamus I'm showing in E. So some of the reasons why this is particularly interesting, the superior colliculus, obviously Patrick Porcelli's group has shown that modulation of the superior colliculus can be a highly effective intervention in stopping seizures in other mouse models of epilepsy. And then the medullary reticular formation, for its importance in modulating things like brain states and stuff like that, we thought perhaps this would be a good potential candidate as a regulator of ictal activity. And then the centrolateral nucleus of the thalamus has recently, or it has been shown before, to be recruited and modulated by limbic seizures, and then also modulation of these neurons in the centrolateral can improve measures of consciousness also in focal epilepsy. So these are three potential candidates, but it's important to note that there are several other downstream regions targeted by the fastigial, including several different thalamic nuclei. These are a few different examples. We have the medial dorsal nucleus, the ventromedial, the ventrolateral, the parafascicular, and zona incerta in the thalamus. We also have several different nuclei in the midbrain, so mesencephalic reticular formation, lateral dorsal tegmental nucleus, and then different regions of the periaqueductal gray. And then we have several different nuclei in the brainstem. So these are two different regions in the pontine reticular formation. We've got widespread projections to vestibular nuclei. And also, as expected, this has been shown in the literature before, but there appear to be fastigial projections traveling to the spinal cord. So this is the more caudal portion of the medulla. In our sagittal slices, you can see that there are these bundles of axons that are appear to be heading towards the spinal cord because they're not terminating in the medulla itself. This is just a subset of all the regions that the fastigial nucleus projects to. In fact, a recent study from Satya Dulac's group looking at divergent fastigial projections found that this nucleus projects to over 60 distinct brain regions. So this nucleus in the cerebellum is really diversely connected with a lot of downstream regions. So if I have one goal today, I would say maybe think twice before chopping off the cerebellum when you're about to do your tissue processing, because this is really an important structure given all of its different interconnections and things like that. So we had to start somewhere. We couldn't test all 60 at once. So we decided to start with these three nuclei. So the superior colliculus, the medullary reticular formation, and the centrolateral nucleus, for those reasons that I mentioned before in terms of their potential relevance to seizure networks. So to target these outputs, we wanted to use an approach where we could really kind of recapitulate as much of that direct vestigial stimulation as we could. So to do this, we used a dual viral strategy to achieve our opsin expression in vestigial neurons that project to a specific downstream target of interest. So with this approach, we inject a virus in our target of interest, so in this case, the superior colliculus, and this red virus is a retrograde virus that induces retrograde expression of Cre. So this will be taken up by axons and induce Cre expression in those cell bodies that project to that area. And so then we follow that by an injection of a Cre-dependent virus in the vestigial nucleus. So this approach will allow us to selectively label just the vestigial neurons that are projecting to the superior colliculus. And then the same when we do our retro injection in the reticular formation, and then also the centrolateral nucleus of the thalamus. So we have these three different populations of neurons that we'll be labeling with this dual viral strategy. So for the vestigial neurons that project to the superior colliculus, I'll refer to these as FNSC neurons. Those that project to the reticular formation, I'll refer to as FNMDV neurons. And those that project to the centrolateral nucleus, I'll refer to as FNCL neurons. So this is what it looks like in the vestigial nucleus with those three different approaches. So again, FNSC throughout this is going to be labeled with purple, FNMDV, this will be labeled with this teal color, and then FNCL will be labeled with yellow. So we can see that rather than getting expression all over the place in the vestigial, we're kind of getting these more restricted populations of neurons. So in the FNSC and FNCL mice, we have populations that are mostly restricted to the more caudal portion of the vestigial nucleus. For the FNMDV, we do get expression some in both the rostral and caudal regions, but it's actually mostly focused towards the more rostral portion of the vestigial nucleus. And what was really cool about looking at the fibers with this is it appears as though this approach is labeling three distinct populations of neurons, so three non-overlapping populations. And the reason why we think that is because if we look at our three different targets of interest, so for example, here with the FNSC neurons, if we look in the superior colliculus, we see nice strong labeling of fibers here, but we don't see much in the reticular formation or in the centrolateral. So we don't see those FNSC neurons do not appear to strongly be projecting to the reticular formation or centrolateral. Same thing for FNMDV. We see really nice strong fibers in the reticular formation, whereas we don't see anything in superior colliculus or centrolateral. And then finally for FNCL, similar story where we see really strong labeling in the centrolateral, but we don't see strong fibers in the other two locations. And this is what the quantification looks like. Basically, we are measuring the length of all of the different fibers in a particular field of view, and we're making these histograms of path lengths. So the shorter path lengths are going to correspond more towards likely correspond with terminals because there's going to be shorter little fibers. So you can see for the superior colliculus, the strongest inputs are really coming from those FNSC neurons. And then reticular formation, strongest contribution is the FNMDV, and then centrolateral, same thing, strongest contribution is from the FNCL. So we think this is really labeling these three different populations of neurons with this approach. What was really interesting though, especially for me as background as a cerebral physiologist, is we weren't just seeing projections to the superior colliculus or reticular formation or centrolateral. So these three different populations also had collaterals in other places. So I'll show you some of those locations in the following slides. So this is looking at those FNSC neurons, so those neurons in the vestigial that are projecting to the superior colliculus, also appear to be projecting to a subset of those thalamic nuclei. So we only see some very sparse expression in the medial dorsal nucleus and the ventral medial nucleus, but we see really strong expression in the ventrolateral nucleus. We only see a couple sparse fibers in the parafascicular, and we don't see anything in zona incerta. In the midbrain, we see relatively sparse, fibers in a couple different midbrain nuclei. So a couple fibers in mesencephalic reticular formation, some fibers in lateral dorsal tegmental nucleus, and some fibers particularly in ventrolateral periaqueductal gray. And in these neurons, we don't see any strong expression in the brainstem nuclei. So nothing in that pontine or medullary reticular formation. And then also we don't see any strong evidence for projections to the spinal cord in this population. So it appears as though these neurons that are projecting to the superior colliculus are also projecting to a restricted group of thalamic nuclei and then some sparse expression in the midbrain. Conversely, with the medullary reticular formation neurons, we see some expression in the thalamus, but again, predominantly in a different region of the thalamus. So the main fibers that we see in the thalamus are mostly in the central portion of that medial dorsal nucleus. So we don't really see much going on, maybe some sparse fibers in ventrolateral and parafascicular in the other thalamic nuclei, but it's really concentrated to the central portion of the medial dorsal nucleus. Looking at the midbrain, we see the strongest expression in the lateral dorsal tegmental nucleus, but predominantly, most of that expression we see dominated by brainstem nuclei. So huge amounts of fibers in pontine reticular formation nuclei, huge amounts of fibers in the vestibular nuclei. However, similar to the first population, we don't really see strong evidence for projections to the spinal cord. So most of the fibers that you see here are actually terminating within the medulla, as we would expect, because this caudal portion of the medulla is really going to be the medullary reticular formation predominantly. So we don't see strong evidence of those same fibers heading to the spinal cord. Finally, for FNCL neurons, similar to the FNSC neurons, these are projecting a lot to thalamic nuclei, but again, it appears to be a somewhat distinct group of thalamic nuclei. So we see fibers in the more lateral portion of the medial dorsal nucleus, but a lot of our expression is focused to the ventral medial nuclei of the thalamus. Only sparse expression observed in the ventrolateral, some expression in parafascicular, and then we also do see a little bit of expression in the zona incerta. So this is one group that does appear to project to the zona incerta. In the midbrain, again, similar to FNSC, we just see some sparse fibers in areas like the mesencephalic reticular formation, lateral dorsal tegmental nucleus, and the more lateral periaqueductal gray, ventrolateral periaqueductal gray. And similar to the FNSC neurons, we don't see any evidence of expression in the brainstem. So these neurons, these FNCL neurons really appear to be heading to a specific group of thalamic nuclei with some smaller collaterals to nuclei in the midbrain. So I showed you a lot of representative images. I'm going to show you the quantification and what that looks like. So this is our thalamic nuclei. So as I mentioned, that more lateral portion of the medial dorsal nucleus is dominated by FNCL, whereas the more central portion is mostly coming from the FNMDV. The ventral medial nucleus is receiving the strongest inputs from the FNCL neurons, whereas the ventrolateral nucleus is receiving the strongest inputs from the FNSC neurons. The parafascicular, we have mostly coming from FNCL, and again, some sparse expression in zona incerta, and it's almost entirely coming from those FNCL neurons. In the midbrain, like I said, we have some areas of sparse and overlapping expression, like in the mesencephalic reticular formation and the ventrolateral periaqueductal gray. But the lateral periaqueductal gray appears to be getting most of its sparse input from the FNCL neurons. The lateral dorsal tegmental nucleus is really getting most of its inputs from the FNMDV neurons. And then of course, the brainstem really dominated by those FNMDV neurons. So the neurons that are projecting from the fastigial nucleus to the reticular formation are also sending really, really heavy collaterals to several different brainstem nuclei. So this is a schematic of some of the more notable regions. So we have our 3 different output channels that I've characterized in these previous panels and figures. So we have FNSC, so fastigial neurons traveling to the superior colliculus, also tend to be sending sparse collaterals to the ventrolateral periaqueductal gray, and then this specific group of thalamic nuclei, medial, dorsal, and ventrolateral. Whereas the FNMDV neurons, so neurons in the fastigial that are traveling to reticular formation, are sending most of their inputs to brainstem nuclei like the vestibular nuclei, medullary and pontine reticular formation. formation. They also have some expression in ventrolateral and periaqueductal gray, lateral dorsal tegmental, and their only input to the thalamus really is most strongly to the central portion of the medial dorsal nucleus. Finally, those FNCL neurons, so neurons in the fastigial that are projecting to the centrolateral, are also sending heavy projections to other thalamic nuclei, but a relatively non-overlapping group from those other two populations. So centrolateral, medial dorsal, ventromedial, parafascicular, and zona incerta, as well as some sparse projections to the lateral periaqueductal gray. So this dual viral approach has identified these three different populations of neurons, so these three different fastigial output channels. And so we wanted to look at whether modulation of either of these three output channels might have an effect on hippocampal seizures. So we used that same dual viral approach, but instead we took our on-demand optogenetics route. So we did our dual viral injection followed by chinic acid and then implantation. So we could do on-demand excitation of each of these three different fastigial output channels. And what we found was really interesting that basically only activation of those FNCL neurons was able to produce a significant effect on hippocampal seizure duration. So neither activation of FNSC neurons nor FNMDV neurons had any effect on the duration of hippocampal seizures, whereas activation of FNCL neurons was actually able to terminate those hippocampal seizure events. This is looking at individual animals. So again, we can see only significant effect is here, where we get pretty good seizure attenuation when we're activating FNCL neurons. And then this is across our population. So we have two different ways we're looking at this. First, we're making a population histogram of those seizure events. So we're taking 100 events from each animal. And then here's the actual normalized duration for each of the mice in the given population. So you can see that the strongest effect is observed with activation of FNCL neurons. And actually, this was a very consistent effect. So 7 out of 7 mice showed a significant effect of light delivery when we were labeling those FNCL neurons, whereas either none of the mice or only one of the mice had a significant effect when targeting the other two populations of neurons. So it really suggests that that fastigial output channel to the centrolateral is highly effective at terminating hippocampal seizures, but that the other two failed to have any effect. So it really is that particular output channel that might be mediating the effect of fastigial modulation. So, do we know if it's just this whole output channel, or is activation of those fibers just in the centrolateral? Is it really that projection to the centrolateral that's underlying the effect that we see? So as a final step, I'll show you some fiber targeting experiments that we did. So we injected a virus for broad expression in the fastigial nucleus, and to activate specifically fastigial terminals in the centrolateral, we instead implanted an optic fiber sitting over the centrolateral nucleus. So the idea is that when we shine our light, we're selectively activating those fastigial terminals in the centrolateral nucleus of the thalamus. So same sort of timeline and experimental schematic. And what was really cool is we found that activation of those centrolateral terminals themselves was sufficient to produce an effect on hippocampal seizure duration. So you can see these events are terminating shortly after delivery of light to fastigial terminals in the centrolateral. We see a significant effect at the individual animal level, and then also at the population level, we see a significant effect in the reduction of hippocampal seizures, with 7 out of 11 animals showing a significant effect of light delivery. So this really suggests that it's that fastigial projection to the centrolateral nucleus of the thalamus that is underlying our seizure control. All right, so what all have I showed you today? First and foremost, I've showed you that two structures within the brain that are not typically associated with one another, the hippocampus and the cerebellum, really have a profound potential interaction. So looking at some of Esther's work that I showed you, where she was targeting neurons in the cerebellar cortex, those Purkinje cells, Esther showed as a postdoc that activation of cerebellar Purkinje cells could robustly attenuate hippocampal seizures. There's not a direct connection between the cerebellar cortex and the hippocampus, so we decided as a first step, my postdoc work, to look immediately downstream at the fastigial nucleus. And so what I showed you in the first half was that the fastigial nucleus has an effect on hippocampal seizures, but the direction of modulation is really critical. So inhibition of fastigial neurons has no effect on hippocampal seizures, whereas excitation of fastigial neurons can really profoundly attenuate hippocampal seizures and thus have a really profound effect on modulating hippocampal dynamics. So I'm using these dashed lines because this is not a direct projection. It's just merely exerting an influence. So we had to look downstream yet again to try to find a potential output that was important. And in the course of that work, I showed you that the fastigial nucleus is really segregated into these distinct output channels. Rather than just having one neuron projecting to one region, it appears to have these segregated populations that are simultaneously influencing multiple downstream structures. And again, just from a basic science perspective, I think this is a highly interesting finding. This concept that you have individual neurons sending the same information to multiple downstream structures of interest. And I think it's going to be a really important and interesting question to pursue further. But in the context of those output channels, I showed you that fastigial projections, particularly to the centrolateral thalamus, are important for that seizure control. So I showed you that activation of the fastigial output channel to the centrolateral thalamus, as well as activation of terminals within the centrolateral thalamus itself, are able to attenuate those hippocampal seizures. So that's kind of the crux of the work that I've showed you so far. Obviously, this is another dashed line, so our work is not complete here. There's not any strong evidence for a direct projection from the centrolateral thalamus to the hippocampus. So one future direction that the lab is taking is from the neuroanatomical route, which is what is the potential intermediate region between the centrolateral thalamus and the hippocampus? What is the route by which those centrolateral neurons can actually, to get to the hippocampus to influence ongoing activity? So that's one future direction that we're exploring, both from the perspective of modulating seizures, but then also how would that potential pathway influence ongoing hippocampal function. Another possibility that the lab is exploring is it might, in addition to a potential neuroanatomical pathway, maybe there's something about both this projection from the fastigial to the centrolateral and the centrolateral itself that is able to control seizures in general. So there's something about this pathway that has the ability to desynchronize or disrupt these highly synchronous seizure events. So one thing that we're looking at is trying to implement these same types of interventions, but in different mouse models of epilepsy. So with seizures arising in regions beyond just the hippocampus. And finally, something that I'm particularly interested in is I've been showing a lot about cerebellum influencing the hippocampus, but we also know that the hippocampus can really strongly influence the cerebellum. And in the context of epilepsy, this has importance, not just from a functional perspective, but also it could be of great clinical relevance. So cerebellar neurons can be activated during seizure events themselves, but then also long-term structural changes, so decreased cerebellar volume, is something that can specifically predict the emergence or predict the occurrence of, it can be more predictive of the occurrence of negative outcomes in epilepsy, like sudden unexplained death in epilepsy. So this chronic relationship between hippocampal seizures and cerebellar activity might have a really high relevance for understanding those potential negative outcomes. So this is something that we're actively working on in terms of examining how hippocampal seizure events and chronic epilepsy influence cerebellar processing. So I'm now moving one step back from the fastigial nucleus back to the cerebellar cortex, trying to develop some novel techniques for characterizing modulation in chronically epileptic animals. And I can't say anything more about that right now. Hopefully next talk or something like that, next post or whatever, I'll be able to share some more information about that. So with that, I would like to acknowledge the following. First, my postdoc advisor, Dr. Esther Crick-Magnuson, members of the Crick-Magnuson lab who contributed to this work. In particular, I want to acknowledge Maddie Tetzlaff for all of her excellent work on the fastigial output channels experiments. So she was involved with a lot of the quantification of those fastigial projections. So I would have been totally lost in fighting with Image J at this point still if it hadn't been for her. And best part is for folks at UC Irvine, you don't even have to take my word for it for how great Maddie is, because Maddie will be joining your ranks in the fall or late summer as a member of the MD PhD program. So keep your eye peeled for Maddie if you're at Irvine. Also, some undergraduates in the lab, Jane Yap and Kayla Togneri, provided technical assistance along with Isaac Hoff. Other members of the Crick-Magnuson lab, and then the University of Minnesota Department of Neuroscience, Mouse Behavior Core, and Optogenetics cores provided a lot of animal assistance and the university imaging centers for all the confocal imaging experiments. And then also our sources of funding, including Esther's funding from NIH and a couple different sources here at the University of Minnesota, as well as the American Epilepsy Society for a postdoctoral fellowship. And finally, I'd like to thank you all for your attention, and I would be happy to take any questions.
Awesome, thanks, Martha. There's this lab at UCI called the Hunt Lab. You can tell Maddie about.
I'll pass that along. I'm not familiar with their work, but I'll check it out.
Yeah, they're not very good, but she might be okay. Anyway, no, that was a great talk. That was a lot of data, and a lot of work, both on the physiology and the neuroanatomy. And so great job there. Rick had a question, so I can jump right into it. We have time for questions. Rick, do you want to ask your question, or do you want me to? I can read it, but she started to talk about the central lateral thalamus a little bit. I'll start reading and you can jump in if I get this wrong. So Rick says, from an anatomical viewpoint, I'd like to see projections that eventually reach hippocampus. From those studied, most likely will be central lateral thalamus, and your selective activation studies demonstrate that. Another strong candidate would be nucleus reunions of thalamus, which has clear projections to hippocampus. Are there any vestigial projections that go there?
Yeah, we have not seen any. Reunions was kind of one of the big ones that we were thinking about, and I have not seen any strong evidence for vestigial projections to the reunions nucleus. Yeah, it's a great question.
So, sort of similar. I was thinking about this too, because you're trying to get to hippocampus, right? That's where the seizures are going off, and there have been a lot of sort of cell type rabies tracing experiments that have looked at basically every cell type in the hippocampus so far. I'm thinking a lot about like Zheng Minzhi's work. He's had a number of papers looking at that. Is there a way to sort of data mine that and compare what they've found in the cell type sort of rabies tracing experiments to what you guys are seeing in your work?
Yeah,
You're trying to build the map. You're trying to build a map, right?
Yeah, for sure. And I mean, I think we're starting to, what some other folks in lab are starting to try to do is do, more of this dual viral strategy, but pairing it with more retrograde injections in the hippocampus to see if we can like identify overlapping populations. So we're trying to get, or I think we have in the lab now, this an anterograde transsynaptic approach to try to get like basically the neurons in the central lateral that are receiving inputs from the vestigial, where are those neurons going? Okay. And then trying to figure out what those intermediate brain regions might be. Right.
So if central lateral is the one, then you may be lucky. But if it's not, then you might be unlucky.
Yeah, definitely.
So yeah, it kind of pops out, right? Because yeah, you're kind of inching there, you're getting close to the hippocampus. That was very interesting. Yeah. Do we have any more questions? Everybody's going to be shy. Okay. Going once, twice. Okay. Well, Martha, this was a really good talk. I thought you did a great job. There's a lot of data here. I'm very impressed with like just the volume of data that you have is really impressive. And thank you very much for giving the talk here. And thank you everybody for attending. Thank you for the entire year. It's been great. And with that, I guess we will close.
Great. Thank you for having me.
Sure. Thank you. See you, everybody.